So this is our last lecture. It's focused on the other way in which the immune system is going to respond to help protect us from diseases. So this is known as the adaptive immune response. And there were several instances during the last lecture where we had touched upon that. And so I'm going to remind you of those instances because we're going to kind of pick up where several of the processes were paused. And so we'll see how that is going to complete. So we're going to start by looking at it in a same kind of overview as we did before. And so this is the same picture that we saw from the innate immune response. And so much of what we saw in that innate immune response focused on the first line of defense, those physical and chemical barriers. So we talked about those. And then part of the second line of defense that also falls under the innate immune response included things like phagocytosis and the complement cascade and interferons and inflammation. What's not included here, we talked about fever. So remember that the innate immune response you are born with, and it is non-specific. So by contrast then, this adaptive immune response is going to be acquired through one's life, and it's going to be specific for particular microbes. So what we mean by that is when you are exposed to a particular microorganism, so let's just say strep throat, so Streptococcus pyogenes, we learned about that in the gram-positive pathogen lecture. So when you are exposed to Streptococcus pyogenes, it is at that time, so during that infection, that your immune system is going to develop protection. So that's what we mean by acquired through exposure throughout one's life. And not only do you develop that exposure at that time, it's going to be unique or tailored or specific to that particular microorganism. And so the protection that develops is not going to be protective against other strep infections or definitely not staph infections or other viruses. And so it's very tailored or specific to that particular microorganism. And so we talked about this before, but there's a group of white blood cells called lymphocytes that are going to be important in driving this adaptive immune response. So if you recall, there is a variety of white blood cells that we talked about, like neutrophils and eosinophils and basophils, and then also monocytes, and they were important in the innate immune response. And so the one group that we haven't really talked much about are called lymphocytes, and they're going to be important in this adaptive immune response. So this also we saw in the last lecture. And so these were the four that I just mentioned, and you can see that they are bracketed under the innate immune response. So if you look at this bracket, it's going to encompass these four. And so the one that we are really interested in that's going to be driving this adaptive immune response are again called lymphocytes. And so those are the ones that we want to better understand. So we'll talk about those first, and then we'll break them down and see how they function and the processes that they are carrying out to help develop this adaptive immune response that we're interested in. Before we get there, there's a variety of characteristics or attributes that we can talk about in regards to this adaptive immune response. So as I mentioned, as you're exposed, you acquire protection through this adaptive immune response, and it's specific. So this is something that is a really complex and actually quite an amazing process. So if you think about what we're saying is your body has the ability to develop unique and specific responses to basically unknown microorganisms that you're going to encounter through your life. So one thing we're trying to figure out is how can your body develop the different cells and processes to allow you to have these specific tailored responses. So that's one attribute we want to better understand. And part of the answer to that is you have a bunch of these lymphocytes, those important adaptive immune cells that are in circulation at all time, but there's really going to be a small subset of them that get turned on. And so the term we use to describe this is inducibility, or they are induced. You can call that activated or turned on. And so you may have this population of lymphocytes, but only a small number of them are going to be induced or turned on. So we want to look at how that process occurs. How can only a subset or certain cells be induced? So we'll get to that. But then once they are induced, what's going to happen? So there's a process that's called basically making a bunch of copies of themselves. So they're generating identical clones or nearly identical clones. And this allows, again, that kind of small number of cells that are turned on. They can fairly rapidly turn into many cells, because they're basically going to make a bunch of clones of themselves. So fancy word is, again, proliferate, which just means divide and increase in number. And so if you just had one cell that's induced, it can turn into 1,000 identical cells in a really quick period of time, so that you have 1,000 cells or 10,000 cells that are fighting the infection instead of just that original one. The other thing that's really critical for the immune system is we want it to attack these foreign microorganisms to protect us, but we don't want it to attack our own cells most of the time. And so we want these immune cells to be unresponsive to our own cells and tissues. And so we have to then have that to be part of the conversation as well, as to how these immune cells can recognize what is foreign and this is what we should attack, versus, oh, this is a normal, healthy human cell, so let's not attack that. So that's another attribute that we want to better understand. And then lastly, immunological memory is a really important part of the adaptive immune response. And so what this means is, after you are exposed to one particular microorganism, not only do you develop protection at that time, that's that acquired part, and then it's going to be specific for that particular infection, but you have some long-lasting, what are called memory cells, that remember what that specific microorganism looks like. And so what that then means is, if there's ever a future encounter with that microorganism, these cells will respond very, very rapidly to generate protection. And often so quickly that you don't even get sick at all, or if you do get sick, it's going to be much less severe and a much shorter duration. And this memory is also what drives vaccines and our vaccination campaigns for a variety of diseases. So that memory is what protects us over a long period of time. That is a really important attribute or characteristic of this adaptive acquired immunity. So lymphocytes are the cells. Lymphocytes are the cells that are going to be important in this process. And so hopefully you may have noticed that they share a root with our lymphatic system. So this word is shared, lymphatic system. And so if you've had your anatomy and physiology class, you might be aware of what this system is. And if not, I'll tell you about it just briefly now. And if you have had this information, this will just be a really quick refresher. And so most people are very much aware that we have a circulatory system that is carrying our blood throughout our body. And so as that blood reaches our tissues at a location called capillaries, there is fluid that leaks out. And so throughout our tissues, we have this excess amount of fluid that's leaking out, that if it were not collected and removed from the tissue, we would just continue to swell and be not looking real nice, because we would just be bloated, and eventually our blood would just continue to leak out our capillaries. So it turns out there's a system, it's called a lymphatic system, that is going to collect that leaked capillary fluid back out. And so the lymphatic system is a series of these vessels. So they're shown here in green. And so they're going to be collecting that fluid within tissues. and then returning it back to the circulatory system at different locations. And so it's going to move through our bodies. So these series of vessels don't have a pump, like it doesn't have a heart like our circulatory system has that helps create a pressure to move the fluid through. And so the way that the fluid is going to be returned is just the muscles as they contract within different areas are going to kind of squeeze it like a tube of toothpaste. And so it's going to be a one-way direction of this fluid that gets ultimately back to the trunk and then dumped back into our circulatory system. So that's one function. So you can think of the lymphatic system as being really important part of kind of a sister to the circulatory system. But what we want to focus on is this one. If you think about what I just described to you, is this lymphatic system is constantly picking up fluid from all the tissues and locations throughout our body. And so it's a really great place to screen for potential pathogens and infections. So for example, if you had a cut right here, on your leg, and you now have microbes invading and causing an infection there, then as you might guess, the fluid that's being collected from that area are going to have some microorganisms associated with it. And so embedded within these series of of lymphatic vessels are going to be these things called lymph nodes, that are going to be important for screening that fluid for the presence of foreign microorganisms, or here's this term again we're going to be using a lot, antigens, foreign antigens, and those are molecules. We'll say foreign molecules from microbes is a good working definition. That's a B there. And so embedded within these lymph nodes are going to have a variety of white blood cells or leukocytes. Included in that can be lymphocytes. We can also have macrophages present there as well. And so that's what these lymph nodes are doing. They have these leukocytes present that are going to be looking for and recognizing and attacking those foreign microorganisms in a way to help protect us. So that's a short version of the lymphatic system. And those lymphocytes that were talked about are what we want to talk about now. And so lymphocytes can be broken down into two categories. So both of these are subtypes of lymphocytes. So they're called B cells, or sometimes they're called B lymphocytes, but usually B cells, easier to say. And then also T cells, or T lymphocytes. So these are the two types of lymphocytes that are present within our body. And so they're both going to be important in this adaptive immune response. As we have said, that's what lymphocytes do. And so that's shared across them. They're both white blood cells, and so they're produced, all blood cells are produced in our bone marrow. So this is true for the other types as well, so lymphocytes included, but so too are monocytes and neutrophils, etc. So what we want to look at then, and what's shown here, are the unique differences that exist, and this is what allows us to say, oh, well, that lymphocyte is a B cell, but this lymphocyte we can classify as a T cell. So while both are undergo their original development in bone marrow, they don't mature until they reach these different destinations. So T cells will migrate to a different organ called the thymus. It may have been on the previous slide. I don't remember if it was present. Yeah. So here's the thymus. So it's located kind of in the center of your chest there. So that's where T cells will migrate to and mature. And so that's where they're ultimately going to differentiate and then get into the circulatory system and then migrate to, they could be in the blood, they could be migrating to those lymph nodes as I was just talking about, whereas B cells are going to mature in the bone marrow. So that's one important difference between them, is where they undergo maturation. A second is a molecule that's on their surface. And this is pretty self-explanatory. The name kind of explains it, but I'll give you a little bit more information. So T cells have a unique molecule. Really, it's molecules. It doesn't have one copy of it, but it has many copies. So that's called T cell receptors. So on a T cell, what you can see is there's this one molecule called a T cell receptor, or TCR for short, that is going to be on the surface of the cell. And B cells have something similar, but it's called a B cell receptor, or BCR, on the surface. So you can see that is across the surface of B cells. And those are important in each of the cell types, so BCR and TCR, because they have the ability to recognize, there's that term again, recognize antigens, as it says, recognize specific antigens. So I'm going to write this definition again. What we are calling, we're going to talk about antigens in a little bit, but again, antigen is going to be, often it is a surface molecule of a microbe. So that could be a bacteria, it could be a virus, it could be a fungus. And so all we're saying is on the surface of a bacterium, for example, there can be a unique molecule that could be an antigen that our immune system might recognize as foreign. And that could trigger an immune response. So we're going to talk more about antigens in a little bit, but we've used this analogy at different times, but you can think of this as very much of a unique complementary relationship, like a lock and a key. And so if a bacterium has this unique molecule on the surface that we can call an antigen, it is possible that our body has a B cell that has a BCR on it that recognizes that antigen specifically. There may be a T cell with a TCR that recognizes that same antigen as well. And so as we will move through this, it's important to realize that both B cells and T cells have these unique molecules, and this is what in part allows for a specific tailored response, because what's going to happen is only the B cell that has a BCR that recognizes an antigen on that microbe are going to be involved in the immune response. And the same is true for a T cell. It has to have a T cell that has a TCR, T cell receptor, that can bind to that antigen. Okay. And if you do get that specific relationship, so a BCR binding to an antigen, then you're going to get a specific response. And B cells are responsible for ultimately turning into or developing into what are called plasma cells. So we'll talk about that later. But what you want to make the connection of, B cells are ultimately going to produce antibodies. So that's the important connection. B cells will ultimately produce antibodies. T cells do not produce antibodies, but instead, the T cells themselves have the effect. So what I mean by that is T cells kill microbes directly, whereas in this case, you have B cells, which are going to make antibodies, and then it's the antibodies that attack the microbe. Okay. So it's just a question of, are the cells doing the direct attack? That's what T cells do. And so we call this the cell-mediated arm, or the cell-mediated immune response. So T cells do that. B cells, because the B cells themselves don't do the attacking, but instead they make this important molecule called antibodies. We call this the humoral mediated response. Okay. And I wrote down here, so I apologize. The other thing that we touched upon is the importance of immunological memory that is generated during this adaptive immune response. So each of these, both T cells and B cells, if they are activated or induced because they have a unique, their unique BCR can interact with a specific antigen, then during that activation or inducibility event, they can generate what are called memory cells that provide that immunological memory, as we talked about. So what I'm going to do is talk about T cells first, just a little bit, but we're going to spend most of our time talking about B cells and antibodies because that's a really important way that our bodies are protected. So a little bit on T cells, and then we're going to move on to B cells pretty quickly. So remember that T cells, they're also called T lymphocytes. So they are a group of lymphocytes that mature in the thymus, as we saw. They have a TCR on the surface, and they are important in what's called the cell-mediated response. So that's what it said on the previous slide. I'm just putting that on here to have, there it is, cell-mediated response. And what that means is the cells can act directly against the microorganism or antigens. They are not making antibodies. Now it gets a little bit more complicated, as you might guess. We are going to talk about 3 different types of T cells. So one are called cytotoxic, and then the other 2 are both helper T cells, but we're going to differentiate between 2 different types of helper T cells. There are more types of T cells in this, but these are the only 3 that we're going to talk about. So these are the 3 that I want you to know. You can ignore the other ones, but these are the 3 important ones. So these T cells that are known as cytotoxic, if you think of that name, cyto means cell and toxic means toxic, to kill. So it is these T cells that have the capacity to directly attack and kill microorganisms. And so this is what's driving the cell-mediated response that we were talking about. These are the 2 types of cells that cytotoxic T cells are going to be targeting primarily. So if you look, it says cells infected with viruses and other intracellular parasites. So these, we're talking about these T cells, these cytotoxic T cells, are going to be attacking human cells, but they're not normal. These are going to be, again, very much abnormal cells because they have viruses inside of them, or they could have other, like bacteria or fungus or some other intracellular parasite. So that just means inside the cell. That's what intracellular means. So that would be one set of abnormal cells that we may want to target and destroy, and indeed cytotoxic T cells can do that. But then another group of abnormal cells are cancer cells. So again, these would be our own cells, again, human cells. These may not necessarily be infected by microorganisms. Some cancers are caused by microbes and viruses, but there's a variety of reasons that cancer cells could develop. But nonetheless, what we're talking about is these are not normal, healthy human cells, but they're abnormal. So those are the population of cells that cytotoxic T cells can target. And one other thing that makes them unique and different from the other types of T cells that we're going to talk about is they have a molecule on their surface that is different. So they have a molecule that's called the CD8 protein, or CD8 cell surface glycoprotein. These are sometimes just called CD8 cells because of that unique molecule on their surface. It does, the CD8 protein on the surface does have a function. We don't go into great enough detail to understand it, but I do want you to know this protein is present on the surface of cytotoxic T cells, and it's not on helper T cells. If you look, there's a different protein on helper T cells. It's called the CD4 protein. And so CD4 is helper T cells are sometimes called CD4 cells. So because that name is pretty consistently used, some people will call them helper T cells, some people call them CD4 cells. So you want to make that connection. CD4 cells equals helper T cells, and CD8 cells are going to be cytotoxic T cells. So we will get to how they function in a little bit, but that's so all lymphocytes mature in the thymus. They all have a T cell receptor on the surface, or actually multiple copies of an identical T cell receptor, and they're involved in the cell mediated response. But then when we break them down, cytotoxic have this additional unique protein. It's called CD8 protein, and these are the cells that are going to directly attack abnormal human cells. The reason we've broken these helper cells down into two types, it's a question of who they're going to help. So they each are gonna, both types of helper T cells have a CD4 protein. Both are going to help by secreting or releasing unique protein signals. They're often called cytokines. And so those cytokines are going to help coordinate and activate this adaptive immune response. But what's different about these two is this last bullet point in each one. It's a question of what those chemicals are going to help. So type 1 helper T cell, their chemical secretions are going to help cytotoxic T cells. That is clearly a misspelling, so I'm going to update that, T cells. And type 2 helper T cells are going to help and regulate B cells. So remember, B cells are going to be responsible for generating antibodies. And so that's indeed, there are some ways of activating B cells that require helper T cells. And so it would be these type 2 helper T cells specifically that are going to drive this production of antibodies. So what we're doing is we're learning about the individual parts, and then we'll put them together to understand the process of how these are going to work. But so far, lymphocytes are important and adaptive. We can talk about two types of lymphocytes, B cells and T cells, so they have their differences. And now we've also broken down T cells into 3 important types. So there's 1, there's 2, there's 3. Okay, so that brings us to B cells and what they can ultimately produce, which is, again, antibodies. I'm going to introduce this to you pretty early on, that antibodies can also be called, I don't know why I can't write there, immunoglobulins, or Ig with some, I'll just put this here, some letter. So for example, you can have something called IgA or IgG or IgM. So there's different letters. So we'll talk about that. But just realize that that Ig stands for immunoglobulin, which is the same thing as an antibody. So that's what we're going to talk about first. And the first thing we're going to look at is, based on this slide, is the structure of antibodies, or what it actually looks like. So an antibody is a protein, and it's a protein, more specifically, that's made up of 4 different protein chains linked together. So remember, proteins are made up of chains of amino acids. And so when we say a tetramer, that means 4. So it's 4 amino acid chains, or 4 protein chains. So there's a heavy chain. That means it's longer, more amino acids. Light chain, that's shorter, less amino acids. And there's 2 of each of those. So 2 identical light chains, 2 identical heavy chains. And the way that they always interact is shown in this picture on the right. And so here is one light chain. Here is the second light chain. Here is one heavy chain. And here, so they're just shown as these lines. So 2 heavy chains. And these S's, if you don't remember from intro bio, we haven't really talked about them. Those are called disulfide bonds or disulfide bridges. And so that's what's holding them. So it's a covalent bond that links this light chain to that heavy chain, and then this heavy chain to that heavy chain, and then this heavy chain to that light chain. So although they are 4 separate chains, they are linked together to form this one larger, more complex structure that we can call an antibody or an immunoglobulin. So now that we have this whole thing put together, we can also look at different regions of it. And so what we can talk about is, hopefully you've noticed, this is kind of how I do the shorthand notation. It's drawn like this, it's like this, it's like that. So it's got a bond there, bond there, bond there, bond there. It looks like the letter Y. So for, rather than drawing this each time to show all four, I frequently would just draw it looking like this. Or if you're really lazy, you just do this. But it looks like a capital Y. And so what we can say is this area where these two arms are, that's indeed called the arm region. This is called the stem. And that area where the two meet is called the hinge. So that's what this is showing up here. There's the arm, there's the hinge, there's the stem. So notice that's the arm where they meets the hinge. And then that's kind of the stem. The most important region that we are going to focus on is at the end of each arm. So we would be talking about right here, right here, or right here, or right here, or right here, or right here. It doesn't really matter. Or in this picture, it's right here. So this is the region right there. That is the antigen binding site. So again, there's that term again. I'll define it again. Foreign surface molecule of microbe. So what we're saying is these antibodies have these little binding sites on the end of each arm that is highly specific and highly tailored to a particular foreign antigen that might be present. So during, again, pick a strep throat infection or a staph infection or a coronavirus infection, each of those pathogenic microorganisms have unique molecules on their surface. We can call them antigens. And if our body is able to produce antibodies that are unique and specific to that each of those antigens, then we can have these antibodies bind to the antigen, and then that can be protective. So this is in the news a lot because they're looking at, if you do get coronavirus, do you have antibodies that are protective for a long period of time post-infection? And then also, obviously, they're looking at the ability of vaccines to generate protective antibodies as part of the response. So anyways, the ability to have these antigen binding sites on the end of each arm is really important. The stem we'll see in a little bit, but we're not going to focus on this too much. But if you remember, there are different types of immunoglobulins. So there's an example, IgG, IgM. And so not every immunoglobulin is just one single Y shape, but you can have something that looks like this. So two Ys, kind of the stems interact. So that's what's called a dimer, two of them. There is another immunoglobulin that has, I don't know if I spaced it out enough. I didn't space it out very well, but it's got 5 immunoglobulins that are, so it kind of looks like a snowflake. Well, that was bad. There's pictures of that later. But that stem region is going to be important for how the different Y shapes interact to form the different types of immunoglobulins. But what we are mostly interested in is understanding every antibody or every immunoglobulin has this shared shape. That is the letter Y. You have a stem, you have the arms, and you have the antigen binding at the end of each arm. So the last piece of the puzzle is shown here in regards to this structure. So remember when we said we have a B cell. So B cell has a thing on its surface called the B cell receptor, or BCR. And so what a BCR is it is an immunoglobulin or an antibody that is actually just tethered or attached to the cell surface. And so I'm going to jump back a couple slides. And this was shown. I didn't make a big deal about it when we saw B cells for the first time, but this is what that's showing. Sorry, I had written over this. And so I'll just jump back real quick. So if you notice this B cell, if you notice that B cell receptor, it's got the shape of a letter Y in there, and that's just to indicate that BCRs are indeed immunoglobulins or antibodies that are just attached to the surface. Okay. So we've talked about antigens. So as I mentioned, it's a foreign substance, or I think I've used the term molecule, so that works too. If you want foreign molecule, I've often said it's a surface molecule. It doesn't have to be, but that's a fairly common location, and it's going to stimulate, or it may stimulate, an immune response. It turns out, and this picture shows it, that the antigen is the whole entire molecule that we're talking about. And it turns out that our immune cells can't bind to and recognize the whole molecule all at once, but instead is going to recognize unique three-dimensional portions of the antigen. So that's shown here. So say this is a nucleus. So here's the cell and here's the surface, and so say this is a fungus within us. And so this is the cell surface of the fungus. And so this thing that's kind of in teal green is the entire antigen. So it's on the surface of this fungal cell. But what actually sticks out are different parts of this antigen that have unique shapes. And so we can call each of those little spikes or balls or whatever that shape is, kind of trapezoid or whatever it is, those would be these unique three-dimensional regions of that antigen that we call epitopes. Okay, so an epitope is a portion of an antigen, and it's going to have a unique three-dimensional shape to it. And this is what our immune cells are actually cueing in on. So even though we've had, for example, antigen binding sites, in reality, so for example, I'll draw an antibody again. So we said on the end of each arm, that is going to be an antigen binding site. It can't bind this entire thing all at once. But we could have, this is going to be hard for me to draw, an antibody that has the ability to recognize this one little region, that specific epitope. So that term becomes kind of equally important into our antigen term. So the antigen is the whole molecule, but the epitope is that specific three-dimensional region that immune cells can and do recognize. So we've talked about antigens a little bit, but then here is just some further clarification and kind of zooming in on how our immune system can be targeting these things. So these two, exogenous versus endogenous, are both from foreign invaders, if you want to call them that. So foreign, so in this case, they're going to be microbes, right? So it could be a virus, it could be a bacteria, it could be a fungus, it could be whatever it is. So there's really going to be two locations that these microbes can exist if they have indeed entered our body. So they can be extracellular microbes, and so that means they're inside our body, but they are between cells or outside of cells. That's what extracellular means. If we're talking about endogenous, this is inside body and inside cells. So think of viruses. They have to enter cells to be successful at making more viruses. There are some bacteria that enter cells as well. And so where that microbe is going to determine what type of antigen that we're going to describe. So in this case, this is a human cell that has a microbe in it. And so here, these are foreign antigens, but they're coming from inside our own cell. Still important to note, but our immune system are going to handle those differently than if they encounter what are called exogenous antigens that are coming from a totally foreign cell. So this could be a bacteria that's just in our gut or in our lungs, or it's in our bloodstream, or it depends on where it is, or just in an infection site because we have a cut. And so these different types of antigens, yes, they're both foreign, but one is coming from inside our own cells, and one is not coming from inside our own cells. And so they're processed and taken care of differently, as we'll see later on. So both of those are important because they're foreign and we may want to get rid of them. And so the last category of antigen is what we call an autoantigen. And these are important because these are the normal molecules that are on our own healthy human cells. And so if you think about this, this is really the set of molecules that our immune system is going to compare everything to. So if these are normal, and we want our immune cells to not attack these normal autoantigens, so that if our immune cells ever discover something that looks different, so say now there's this unique molecule here that is a viral protein that's on the surface of our own cell, or if this bacteria is present, so here's a bacterial protein that's on the surface, or here's a flagella that has its own unique antigens on it as well. Those foreign antigens, whether they're exogenous or endogenous, may trigger an immune response and will then therefore be targeted for destruction. So this is normal, and these are foreign. And so to define what is foreign, we need to have a normal set of antigens. And so that's indeed what autoantigens are. Okay. So what this is showing us is, hopefully you see there's one arm of an antibody. We don't see the whole stem, but here's another arm. At the end of each arm, as we have said, there is an antigen binding site. This doesn't show the entire antigen, but it's got its own, unique shape. I don't know. We'll just draw some weirdness up here. Okay, so say that's the whole antigen. Notice that this antibody does not bind the entire antigen all at once, but instead, there's only this one three-dimensional region, 3D region of antigen. And so, of course, that has a name. We defined it earlier. That three-dimensional region is called an epitope. And so what this is showing us is this one antibody can only recognize this one epitope of this one antigen. So it's got a lot of terminology, so I'll say that again. This one antibody has an antigen binding site that will only recognize this epitope of this whole antigen. And so if you look at that same, here's just the same arm again. Here's the same antigen binding site. So this is a different antigen that doesn't fit. Here is another antigen that it fits, but it doesn't fit perfectly. And so this is again where the lock and key sort of interaction is a helpful analogy, because this is the only instance where the antigen binding site fits the antigen perfectly. And so this, not only does it fit, but it'll activate, the lock, so to speak, and you're going to get a response here. So we're going to talk about those responses in a little bit. But these two, you wouldn't get any response, at least from this one antibody. This antibody would do nothing for this antigen. This antibody would do nothing for that antigen. Okay. So as this says, a single antibody, which is what we have here, only has an antigen binding site that can recognize a specific epitope of a specific antigen. Okay. Now, it is possible that our body does generate more than one antibody, obviously. And so a different antibody may be perfectly capable of recognizing a different epitope of a same antigen. So for example, this unique shape up here is a different epitope. So it may very well be possible that we could generate an antibody that recognizes this epitope of the same antigen. Okay. So our immune system is recognizing antigens, but it's really at the level of these three-dimensional binding sites that are called epitopes. It also might be possible that we can generate an antibody that recognizes this epitope of this antigen, but this antibody isn't capable of doing it. So we were talking about that this adaptive immune response is really important in driving the specificity of this. And so this is in part showing you how we achieve that specificity. So this antibody will be very specific to this epitope of this specific antigen. So again, if this were, say, on the surface of a staph infection, these antibodies would be really important for fighting this specific strain of Staphylococcus. But it wouldn't be helpful to fight other staph or definitely strep or viruses or whatever. So very specific, very tailored, which is one of the attributes that we were introducing earlier. So antibodies, as we said before, have this characteristic shape. So that's kind of the shorthand version of it. And so you can see there's the antigen binding site that we were talking about at the end of each arm. You don't have to memorize this, but I just wanted to put it up there to show you that not every antibody is exactly the same. So there's that kind of naming scheme that IG stands for immunoglobulin or antibody. And then we attach a letter like A or D or E, etc. And so what's different about them is how those letter Y's aggregate together. So for example, here is again, it's called a dimer, where you have two back to back, or this is called a pentamer, where there are 5 that are interacting. So what antibody has what shape isn't overly important, but that's in part what differs. And so the only one that I really want to focus on, and it's really for a couple reasons, is this one. So IgG. And the reason why this one is so important is it makes up most of what is in your circulation. So if you're talking about antibodies in somebody's blood, it's mostly IgG. And the other thing that's important is IgG is going to exhibit all of the functions that we're interested in. So not every immunoglobulin has all the functions, but IgG is pretty good at doing everything that antibodies can do. And so rather than learning what are the functions of each individual one, if we understand IgG is the most abundant and it can carry out all of the important processes that we're interested in, then that's a good one to focus on. So that's indeed what we're going to do. And so this next slide is going to list basically these. There are 4 functions that we're going to talk about that are here. So it's called complement, activation, agglutination, opsonization, and neutralization. So we'll remind you, most of those we have defined previously, but I'm going to remind you of them here. So the first, there we go. So one function is complement activation. So if you go back to the innate immune response and look at the complement slides, what you're going to see is complement are a series of proteins that are not active when they're made. So another way of saying that is must become activated. So there's a variety of ways that this can be accomplished. This activation can be accomplished. We discussed two at that time, and one of them was called the classical pathway of activation. And what that involved is antibodies, so there's that letter Y again, are going to bind to the actual microorganism. In this case, it's a bacteria. And that's going to trigger the series of events that activates all the complement. I'm not going to review it now, but there's another pathway that's called the alternate pathway that does not require antibodies, but instead requires molecules on the surface of the bacteria to be present. And so activating complement through the classical pathway is one thing that antibodies can do. And then if you look back, you can see why are activated complement helpful. So if you go back and look, there's a variety of things that activated complement do. Or I guess I can change that to how are activated complement helpful. One is shown here. These are the membrane attack complexes, or MACs, that form in the surface of these bacterial cells that form holes. That's what the membrane attack complex does. And so by activated complement, you can get lysing of bacteria. So that's beneficial, but there's other things that complement does as well. So I just refer you to your notes about that. Second, opsonization. So as you can see here, each of these bacterial cells has an antibody attached to it. And what we mean by that, if you were to zoom in, right, here's the bacteria. They have these unique molecules on the surface, again called antigens. And so if we have an antibody that fits perfectly, so there's the antigen binding site, we now have basically a nice handle for phagocyte to grab. So if you go back and look at the steps of phagocytosis, there are 5 of them, and the acronym is CAKE. So this first one is chemotaxis, but this second one is what we're talking about. This is adherence. So anything that we can make it easier for our phagocytic cells. So in this particular example, it says macrophage. And that's usually the one that does most of the ones with antibodies attached. But nonetheless, that stem region is a easy place for immune cells to grab onto for this adherence step. And then the next one, if you don't remember, is ingestion and then killing. So being able to grab on is helpful. So we can say antibodies serve as opsonins. Those are those molecular tags or highlights or labels or whatever you want to call it. In this case, they have nice little handles to grab onto, and it makes phagocytosis more efficient. As this says, it's going to be enhanced. Okay. Next, neutralization. So here are 2 examples. This is a flashback. In this case, we have an enveloped virus. So it's got a capsid. That's what that little blue thing is. It's got its nucleic acid on the inside. And then it has an envelope around it, and it's got these viral spikes that project out. And so if you think back, excuse me, to the importance of what these viral spikes are designed to do is those are important keys to enter the locks on human cells. And so what we're showing here is those antibodies, you can see them surrounding the entire surface. So they're basically going to come and kind of gum up all of those spikes, or we can use the term neutralize this virus so it cannot infect another cell. So that's a really important process to help prevent the spread of the infection. So that's neutralization of a virus. So antibodies block the binding sites of the virus. Or we also saw in our infectious disease and our bacteria lecture that toxins are another really important way that bacteria can harm us, whether that's an enterotoxin of the gut or a cytolytic toxin that can destroy our cells, etc. Those toxins are going to bind to specific cells and disrupt their function or potentially kill the cell. And so in green here, this is the actual exotoxin. And so it's possible that we can have these antibodies binding to the exotoxin so this can no longer bind cell, and so that makes it no longer toxic. And so that toxin has been neutralized, as that term is. So that's an important function as well. And that's not all. There's a fourth function that we can talk about that's called agglutination, and that's shown here in this picture. And so again, if this is a bacterial cell, this is a second bacterial cell. Because each antibody has two antigen binding sites, one at the end of each arm, what you can see here is this one antibody is not only bound to this bacterial cell, but it's also tethering this bacteria to it. And then another antibody is linking this cell to this cell. And so you're getting this aggregation. And so that does several things. These bacteria are basically stuck here. They can't necessarily invade. So if they are trying to spread throughout their body, they're stuck here. So that's called agglutination. And it also makes phagocytosis more efficient. And so the analogy that I like to use here is if you're sweeping up a room, it makes sense to sweep up different regions into nice little dirt piles that you can then use your dustpan to pick up. It doesn't make sense to have the dustpan collect every square inch of the room, but if you can sweep to common areas, like in this case, if you can aggregate bacteria to common areas, then when a phagocyte comes, it can be very efficient because it can eat these more quickly and get rid of them. Also, there's a possibility that our spleen filters out things, and so filters work better at taking out large things, and so if they're aggregated together, filtration can be more efficient.